Hello, welcome to this workshop, which is not really a workshop. Anyway, in this presentation, I will bring you to some basics of genetic testing and then apply these basics to different genetic disorders. What is genetic testing? Genetic testing is a type of medical test that identifies changes in chromosomes, genes, or proteins. And these tests are to find changes that are associated with inherited disorders. They can be under medical care to establish the diagnosis in a symptomatic person, or for laboratories as a predictive testing. They can also be requested through a personal decision-making, predictive, carrier, or prenatal testing. So we have mainly three types of genetics: diagnostic, predictive, and carrier tests. The diagnostic test is used to rule out a known suspected disease in a symptomatic individual. The predictive test determines whether an individual will develop the disease before any symptoms appear. And the test is called if the development of the disease is certain when the mutation is present, for example, in Huntington's disease, and may be predispositional if the development of the disease is possible, such as in brain, for example. Carrier testing is offered to individuals with family members with genetic condition, family members of an identified carrier, and if both parents of the test can provide information about what a couple's risk of having a child with a genetic condition. And this test is provided for disorders inherited in an autosomal recessive or X-linked recessive. So the result of testing is to confirm or rule out the suspected genetic, or help determine a person's risk of developing or passing on a genetic disorder. It has always potential benefits, whether the result is positive or negative for gene mutation. For example, a negative result can eliminate the need for unnecessary checkups and tests, and a positive result can direct a person towards available prevention and treatment options. Additionally, some test results help people make decisions of having children. And further, newborn screening can identify genetic disorders early in life, so can be started as early as possible. A positive result means that the lab found a gene mutation, and depending on the purpose of the test, the result may confirm that or indicate that a person is a carrier of a specific mutation, or identify increased risk of developing a disease in the future, or suggest a need for further testing. And positive test result may also have implications for certain relatives of the person testing. A negative result for specific genes means that the lab did not see mutation and indicate that the person is not affected by the disease or is for a specific genetic mutation or does not have an increased risk of developing the disease. So, however, that the test missed a disease-causing mutation because many tests cannot all genetic changes that can cause a particular disorder. Hence, further testing may be required to confirm results. If a person is to be diagnosed with a genetic condition, the genetics professional will be provided with information about the clinical diagnosis, family history that could indicate how the is inherited and the chance of passing it to future generations, and the suggested testing treatment. Three modes of inheritance of the three modes of a genetic condition are dominant inheritance, where one copy of the gene is dominant over the other. Recessive inheritance, where the gene is expressed only when both copies are the same, and a linked inheritance, where the mutation is on chromosome X. An autosomal recessive disorder replicated copies of a specific gene in order to develop one from each parent. And the two carriers parents carry one copy of the mutated gene, have with each pregnancy a 20% risk of having an affected child with two normal genes, 50% risk of having an unaffected child who also carrier, and 25% risk of having an affected child with two recessive genes. In an autosomal dominant disorder, the mutated gene is a dominant gene. So the unmutated copy from one affected parent to be affected. And for such a couple, this means that with each there is 50% risk that we will have a child who inherits both copies of the working gene, and in the child will be unaffected by the condition. And 50% risk that they will have who inherits a copy of the dominant gene mutation and the working copy. And so will be affected or disposed to developing the condition caused by the gene mutation. What you have to know is that in autosomal dominant disorders, if the mutation always produces the disease is called fully penetrant. And if the mutation produces its effect less frequently, 100% of the time, it's called incompletely penetrant. So the penetrance refers to the proportion of people with a specific mutation in a specific gene who also show symptoms of genetic disorders. If some people with the mutation do not develop features of the disorder, the condition is said to have or incomplete penetrance. For example, we can say that the penetrance is 80%. People with a mutation in the gene have 80% lifetime risk of developing the disease. On the other hand, myotonic dystrophy, for example, is an autosomal dominant disease with a complete penetrance. That means that every mutation in the gene will show symptoms. So an incomplete penetrance is associated with autosomal dominant inheritance and never with recessive inheritance. Finally, linked disorder are caused by mutations in genes on the X chromosomes. So a male carrying a mutation will be affected because he carries only one X chromosome, and a female carrying a mutation in one gene on the other X chromosome is generally unaffected. For X recessive disease, as in autosomal recessive inheritance, 2 copies of a disease allele of the chromosome X are needed for a female to be affected, but only one for a male. Thus, female are usually carriers because they only have 1 copy of the disease allele. And for carrier female with pregnancy, there is 1 in 2 chance her sons will inherit the disease allele, and 1 in 2 chance daughters will be carrier. And women are affected when they have 2 copies of the disease allele. In this case, all of her sons will be affected, and all of her daughters will be unaffected carriers. And since males are hemizygous for X-linked genes, any male with an X-linked recessive disease allele is affected. So affected males transmit the disease allele to all of them who are then carriers, but to none of their sons. Or to some, the X-linked recessive usually occurs in males. A single recessive gene on X will cause the disease. No male-to-male transmission, while an affected mother passes the mutation to half of her daughters and of her sons. Now that we know what is genetic testing and how a genetic condition is inherited, what kinds of gene mutation are possible. To function correctly, each cell on thousands of proteins to do their jobs in the right places at the right times. And by changing a gene's instruction for making a protein, mutation can cause the protein malfunction or to be missing entirely. And when a mutation alters a protein that plays a critical role in the body, it can cause a medical condition. And a condition caused by mutation in one genes lead to genetic disorder. The types of mutation that mostly affect the DNA sequence of a gene include deletions, substitutions, and insertions. Changes the number of DNA bases by removing a piece of DNA. Small deletions remove 1 or few bases within a gene, while a larger deletion can remove an entire or several neighboring genes. So the deleted DNA may alter the function of a certain protein. An insertion changes the number of DNA bases in the gene by an extra DNA into the existing gene. And as a result, the protein made by the gene may not function properly. A substitution is a change in one DNA base pair that in the substitution of one amino acid for another in the protein. As we said before, gene mutations may alter the function of essential proteins, and these changes at the protein level include missense mutations, nonsense mutations, and frame shift mutations. A missense mutation is the result of the substitution of one amino acid, another in the protein, which can result in a significant change to the protein. A nonsense, or its synonym, a stop mutation, is a change in DNA that causes a protein to end its translation earlier than expected. It's the substitution of a single base to the appearance of a stop codon instead of the original codon specifying an amino acid. And the presence of this premature stop codon results in the production of a shortened and non-functional protein. And finally, a frame shift mutation occurs when the addition of DNA bases changes a gene's reading frame. A reading frame consists of a group of bases that each code for one amino acid, and the frame shift mutates the grouping of these bases and changes the code for amino acid, and the resulting protein non-function. So insertions, deletions, and duplication can all be frame shift. One technique used to test for genetic disorders is DNA sequencing, which determines the order of bases in an autosomal and genetic code. So I will focus on the original sequencing technology called sequencing, which was a breakthrough that helped scientists determine the human. And next generation sequencing will be discussed later on with Professor Massimo, I think, tomorrow's session. So the first step is to extract DNA from blood. So the DNA purified and amplified by a PCR technique. And the purpose of a PCR is to make a number of copies of the gene, and this is necessary to have enough starting template for sequencing. Then the sample obtained is placed into an automated sequencer. The DNA sequence is then compared to a reference sequence. In the second presentation, I will speak about applying these basics to different types of disorders. Disorders caused by a specific point mutation, like ataxia with vitamin E deficiency, LRRK2 related Parkinson's disease, and these disorders will be detected by gene sequencing. And I will discuss also disorder caused by exons rearrangement, such as muscular dystrophy, which will be detected by another technique, the MLPA technique. In the case of ataxia with vitamin E deficiency, genetic testing in suspected patients is because The treatment for AVID is lifelong high-dose oral vitamin E supplementation, plasma vitamin E concentration to normal level. So if the supplementation is initiated in the early stage of disease, ataxia and mental deterioration may be reversed. And if initiated pre-symptomatic individuals, these individuals may not develop the symptoms at all. Molecular analysis of the TTPA gene, the only gene known to be associated with this disease, leads to the confirmation of the diagnosis by detecting the presence of a gene mutation. Many mutations have been reported in the gene, but the 744 mutation represents the most common pathological mutation in Tunisia, likely because this mutation is a deletion of NA base at position 744 to the TTPA gene sequence. And the diagnostic test consists in the amplification of exon 5 to screen for the mutation. Sequencing this exon will generate different parallels depending on the genetic status of the patient. As you know, it is inherited in an autosomal recessive manner, meaning that an affected child needs two mutated genes and carriers are asymptomatic. So in this slide, we can see the sequence of an affected child with the presence of NA base at position 44. And this is because the child got one normal copy of the gene father and one other normal copy from his mother. And here is a sequence of an affected child where instead of the normal two consecutive A's, have an abnormal sequence with a deleted A. Hence, the child inherited two A's, one from each carrier parent. Another example is LARC2 related disease, which is an autosomal dominant disease. So the LARC2 gene has been as a disease susceptibility gene for PD with G2019S mutation at the bottom or 6055 G2A mutation at the DNA level being the most frequent. And this mutation reported in both familial and sporadic cases occurs at in a North African population and has been reported in 42% of Tunisian families with suggesting founding haplotype in Tunisian patients. So the genetic test used in our lab for the diagnosis of the LARC2 related PD Tunisian patients is mainly the screening of the G2A mutation by direct sequencing exon 41. In this slide, the sequence of an unaffected child with a normal G at position 60. And this is because the child takes two normal chromosomes from each parent. And here, the DNA sequence of an affected child, where we can see an overlay of the G and the A base compared to normal sequence. And this is because the patient is heterozygous, takes one normal recessive gene with a G from one parent, and one non-latent gene with an A from the other parent. A patient may also be with the presence of two mutated copy genes, and hence the DNA sequence shows the presence of an A base instead of a normal G base at position 6055. Other than disorders caused by point mutations, as one I just, we have disorders caused by exons rearrangements, which are large genomic deletions or mutations. And one of these diseases is Duchenne muscular dystrophy. And used is no more direct sequencing, but MLPA, which stands for Complex Ligation Dependent Assay. It's a PCR assay that uses different probes, each specific exons, to evaluate the relative copy number of each DNA. So each peak of fluorescence represents a specific exon, and different relative peak heights indicate a copy number change of the probe target sequence compared to a control sample. And deletions are clearly observed by the absence of the specific peaks for the target gene. In DMD, mutations that lead to lack of dystrophin tend to cause the disease. So the most prevalent mutation occurring for 65% of patients is the deletion of one or more exons of DMD gene. So the DMD MLPA analysis is based on the use of two kits to detect all 9 exons of the gene. And here is a profile with a ratio between 0.75 and 1, indicating that we have a healthy individual. Whereas a ratio less than 0.75 stands for heterozygous or homozygous deletion, and over 1.3 duplication. Here we can see the profile of a young boy who shows, compared to normal control, the deletions of exons 46 to 50, confirming the diagnosis of DMD. And after we normalize the previous profile by software controlizer, we get these histograms where we can see the multiple exons deletions for the child and the carrier status of his mother. Finally here, a boy with of exon 18. Indeed, the ratio is greater than 1.3. And there, because duplications in DMD patients are less frequent and occur in about 6 to 8% of patients. So to conclude, I presented presented you with the Sanger sequencing technique and MLPA to detect genetic mutation. It's important to note that the technology evolved, and now we have NGS, which stands for generation sequencing. And NGS is similar to Sanger sequencing. You sequence DNA fragments, but in NGS, it's massively parallel, meaning that of DNA fragments are sequenced in a single run, whereas Sanger sequencing only put one forward and reverse read. So here is what research to consider. With its ability for long reads and gold standard accuracy, sequencing is perfect for sequencing single genes, for sequencing 100 targets at the lowest cost, and also for NGS confirmation. While next generation sequencing is faster, cheaper, and perfect for screening over 100 genes at a time, and also finding novel variants by expanding the number of targets in a single run. So thanks to NGS, we have now two methods, whole exome and whole genome sequencing, which are increasingly used in healthcare and research to identify genetic variation. So this will be presented in more detail by Professor Masoudi, as I said before. Thank you for your attention.